Haley Streff. Haley is a certified genetic counselor at Texas Children's Hospital and Baylor College of Medicine. She completed her master's degree in genetic counseling at the University of Texas Health Sciences Center in Houston. She sees patients in the outpatient genetics clinic and multidisciplinary craniofacial clinics at Texas Children's Hospital. She also works on genetic test utilization at TCH and is a clinical supervisor for the Baylor Genetic Counseling Program. Haley enjoys participating in outreach and education in the rare disease community, and she serves on the board of directors in multiple committees for various genetic and family support groups. So, thank you.
Okay. Oh, thanks, guys. Okay, so thank you so much. Again, my name is Haley. I'm a clinical genetic counselor. I'm really thankful and excited to be here today. I was here at the family conference back in 2019. I did childcare for that. So it's actually really cool to see how you guys have grown, and I'm really excited to be able to present today. I work with, if you guys know Dr. Wengler, I work with him sometimes at Texas Children's. Part of my time is spent seeing patients, evaluating patients. I have diagnosed some patients with EBF3 or HADDS before, and I spend a lot of time diagnosing kids with other types of rare diseases. And so I think as a genetic counselor, one of my favorite things is that I get to have these kind of cool, close relationships with the families. I've attended lots of other different types of genetic conditions or syndromes, their family conferences. This is one of my favorite things I get to do, this type of outreach. So I'm really thankful you guys had me today. In general, I left a lot of time for questions. My goal today is kind of give you that foundational knowledge or that reminder of your foundational knowledge of genetics and leave a lot of time for questions. So there's a few things I want you to walk away with, but then I want to answer your questions. When we get there also, I will be around if you have very specific questions, or Dr. Wengler also can answer questions too. Anything I can answer, he should be able to answer as well. So to kind of jump into it. So these are my goals. So like we said, I do work training future genetic counselors, residents, fellows. And one of the things I always tell them, especially at an appointment where we're going to be talking about a new diagnosis or results, I say, what are the 3 big takeaways? What are the 3 things we need this family to walk out of the room understanding? Of course, we need to answer their questions, but then what are our 3 goals? So for me today, my 3 goals are to kind of give you that basic reminder of molecular genetics. This is something you probably touched on in high school biology, which may have been a long time ago. And if you didn't go into the sciences, we're going to give you that reminder today. We're going to talk about inheritance patterns and recurrence risk. And then I also am going to introduce some information about types of genetic variants and different sequencing technologies or how you come to a diagnosis. And then, of course, leave time for your questions. Like Ashley said, also, it's the morning, and I don't like sitting in talks like this for a really, really long time or all day. So like stand up, stretch. There's a beautiful view behind you. Go look out the window for a couple minutes if you need to. Just take a breather. That is something I wish I did more often when I'm in talks like this. Okay, so we kind of saw this earlier, but this is like where we begin. Our bodies were made of billions of cells. Inside of our cells is in the nucleus is where our DNA is. When we're talking about DNA, we're really talking about our chromosomes. Our chromosomes are simply the structures that hold our genes. We have thousands and thousands of genes. And really the purpose of a gene is it's basically a sequence that codes for a protein. And I use the word protein as like a small molecule, essentially, not like a steak. That's a little bit different. There are like we think of food protein, but this is like at the molecular level. So when I use protein, protein is simply something that is inside the cell and goes off to do jobs. It binds to things, it sends messages between cells. So it's a function of a cell in human biology. That's kind of what we mean when we say protein. And proteins exist because they get their instructions from genes. So genes are kind of like your body's instruction manual. We have thousands of them. We know where most of them are, but we don't necessarily understand the function of all of them. So you maybe have seen something like this before. This is kind of a cartoon drawing of chromosomes or a karyotype. So humans, we typically expect them to have 46 chromosomes. You have half from the one parent and half from the other parent. So the first 22 are we call autosomes, and then so they're just numbered 1 through 22. They're actually numbered largest to smallest. So number 1, chromosome 1 is bigger than chromosome 22. And then the last set of chromosomes are your sex chromosomes. Females, we typically expect to have XX and males XY. So when we talk about chromosomes and genes, this is kind of what we're thinking about at a genetic level. The EBF3 gene is located right there on chromosome 10 at Q26.3, and that's really just the naming technology us in genetics use to know where exactly on chromosome 10 that gene is located. So you can see it's at the far end of one side of chromosome 10. Everyone's with me. It seems like good nods. Okay, I'm happy about that. So the other thing to remember is, again, we have 2 copies of every single chromosome. So we have 2 copies of every single gene in our bodies. You get 1 copy from one parent, 1 copy from the other parent. And there's a couple of different ways. So when we think about HADDS, we consider a dominant disorder. And that means that if 1 copy of that gene is not functioning how we think it should or how it needs to, that person has that genetic condition. Even though you have that other copy of that gene that's presumably working fine, that's not sufficient to recover that function. So that's what we consider a dominant disorder, and that's what we're talking about with HADDS. It's considered a dominant disorder. So having that 1 copy, just one of your copies not working how we need it to, that's when you would have HADDS. There's other types of genetic disorders, we call them recessive, where you have to have both of those copies not working. So sometimes that gets confusing, but for us, remember, it's a dominant disorder. And when we think about dominant disorders, that means that this kind of gets into your inheritance pattern. So a person who has a dominant disorder, 1 copy of that gene's not working properly, the other one working fine. So when that person goes to have a child, it's a 50-50 chance which copy of that gene you're going to pass on, which chromosome 10 are you going to pass to your future child. There's no way for it's something that it's chance. Every single time you have a child, it's a flip of the coin. So it's a 50% chance in every single pregnancy. So even if you have your first child that maybe didn't inherit that, same 50-50 chance in your next child. Everyone's with me? Okay, good. You guys are doing a good job giving me feedback. I appreciate that. So this is kind of what we just talked about. Autosomal dominant disorder, 50% chance for a person with HADDS to then have their own child with HADDS. And then we put in this other thing about there's up to a 50% chance for a person who's mosaic for EBF3 HADDS to have a child with it. And mosaicism may be something some of you have heard of, but not all of you, but it is something that has been reported in this condition. So I'm going to touch on it. So I tried really hard to find a better kind of visual representation of mosaicism. And this is where I landed. I looked at a lot of images and this is where we landed. So mosaicism is essentially the term we use to say that not every cell in your body or not every copy of that gene or chromosome has that genetic change. Only some of them do. So that would be like you get a genetic blood test and only some of the blood cells carry that genetic change or have it. Let's say only 20% have it, but the rest of those 80% don't have that genetic change. So that's when we use mosaic. That's kind of the term. So this is usually something that happens very early on in embryonic development, because we all start as one single embryo and then that replicates lots and lots of times. And what will happen is that somewhere in that replication process, one of those replications, that replication will be where that variant or that genetic change happens. So you have one that's affected and none of the other cells are. And that one then will perpetuate that to the ones that replicates into. So early, and also when you're early in embryonic development, it's pretty easy to be like, these cells are going to go off and make the skin, and these cells are going to go make the brain, and these cells are going to make something else. So that's why different types of tissues can have different genetic components. So I don't like this photo because it's not like your left arm's affected and your right arm's not. That's not quite correct, but there's not a good way to visually show that. But for instance, you could have a very low amount of cells affected in your blood, but a much higher affected if we did a skin biopsy. So it's hard to tell exactly what percent mosaicism you'll be somewhere, but it is a phenomenon we've observed in this condition. And it's one of those things, it's something we can never fully rule out because we can't test every single tissue in a person's body to tell you that it's definitely not in every single tissue. So that's why whenever we talk about recurrence risks, a geneticist is never going to tell you this will never happen again, because we can't check all of the cells. We can't say with absolute confidence, you don't have this genetic change anywhere in your body. In general, typically the lower level of mosaicism you have, the fewer symptoms you're going to observe in that person too. Are we all still with me? Okay, good. This is That makes sense. So again, since we can't tell how high someone's mosaicism is, we say it's still up to that 50% chance. And one of the reasons we do that is because you can't test, for instance, a person's eggs. There's no way for me to go in and say how many of these eggs are affected. That's not something we have the ability to do. So that's why the risk is always going to be up to 50%.
How do you even determine if someone has mosaicism, or is this just theoretically this is what is
Yeah.
occurring?
So when we're talking about like, there's kind of two different types. So one of them we would consider to be somatic. So that would be, let's say a parent, for instance, gets a blood draw because we know that their child has HADS. We want to see if it was inherited. And that blood draw comes back and says it was 20% mosaic. Well, then we know that person has at least 20% mosaicism in their blood. So we can do it. We can diagnose it that way. But there's always going to be something called germline mosaicism, which is essentially when the gonads are affected, but no other testable tissue types are. And that's because in very early cell development, the cells that go on to become the gonads, the eggs and the sperm, kind of break off pretty early in the developmental process. And again, there's no way to test all of them. Once they're in your body, you can't continuously test those. So it's rare, but it does happen. And so again, we can't ever say zero, but we can say very low. If you have a normal blood test, for instance, or a normal cheek swab, that's very reassuring. Most likely you're not mosaic, but we can't say that with absolute certainty. And that's why there are reproductive technologies, including things like IVF, PGT, which is pre-genetic testing with molecular testing, and prenatal diagnosis. So there are things we can do. We can test embryos, for instance, to see if they're affected. And we could do things, we could do a test while a person is pregnant to see if that child or that fetus that's being carried is affected. So there are things that we can do to kind of understand the risk for a particular couple in a particular situation. I don't have a lot of information about this in my talk, but if someone's interested in, or if you guys have more questions about it, we can absolutely talk about it a little bit more at the end. Any other questions? Yeah. I can repeat it. Mm-hmm.
What? What if a couple with HADS had a baby? Like, what is the recurrence risk?
So you're saying if a person with HADS has a baby, what the chance that baby will have HADS?
No, what if a male with HADS and a female with HADS had a baby?
Okay.
What is the recurrence risk?
Yeah, so that it's going to be, the recurrence risk is probably going to be up to, we're going to say 66%.
Okay.
There's a few other things you have to consider when you do that, because there's a higher chance that there's 50% chance the person with HADS will pass that on and the other partner will pass it on. So that increases that risk. And a lot of times in a dominant disorder, it's a person cannot have both copies not working. That's typically not a viable pregnancy. So it's usually not, the numbers change a little bit. So it's closer to 66%. Or 2/3.
So what would that be called? Would that be dominant or?
It's a dominant disorder either way. It's still dominant inheritance. We can talk a little bit more about it, and I can kind of draw it out for you, which I think might help.
Yeah.
Yeah, we can talk about that, or I can draw it out for you. Good question.
There was and you may have already covered this, there was a question that came in from the Kosman family virtually. How would you find out if mosaicism is applicable to your child? Is it a special test?
So again, typically parents are tested after a child or is diagnosed or concurrently with the child. And so if a parent is tested at the time and that test is normal, it's very reassuring that you are not mosaic. We have a whole team of prenatal genetic counselors at most places. I mean, you can also see them virtually, so you can always talk to a prenatal genetic counselor to kind of talk through your specific recurrence risk skin. I talked about mosaicism. I know it can kind of be an overwhelming, scary thing. It's not the norm. Most people aren't mosaic that we've identified, at least for HADS, but there have been a few identified. But again, you should be reassured if you had a normal test as a parent. But again, we can't say with 100% certainty you're not mosaic somewhere. Okay, so we're gonna shift gears a little bit to talking a little bit more about genetic variants, or exactly what we mean when we talk about variants. This says mutation. We usually don't say mutation anymore. We say pathogenic variant is kind of the scientific term, but I've been using these Greenwood handouts for a very long time. So again, if you think about chromosomes, your chromosomes are kind of like an encyclopedia set. That's another way, this is a common analogy. So you have 46 chromosomes, you have 46 encyclopedias. Your genes are like chapters or topics inside of those encyclopedias. And so when we're talking about a gene, we're talking about reading through an entire chapter, and we're looking for things like a missing word. We're looking for things like a misspelled word that would make that chapter, that sentence not make sense anymore. And that would be equivalent to a gene not functioning how we need it to. Genetic variants are sometimes a little fuzzy to interpret though. So whenever you do like a lab report, so a lot of you, most I would say families in here have experienced genetic testing. And there's a couple of things that can come back from that genetic test report. So we can say, basically the lab's gonna put it into 5 buckets. They're gonna say this is definitely benign, meaning that this is just normal variation. It's one of the many things that make you unique or your family unique. It's not going to result in any medical complications or differences. They can say it's likely benign also. We're not really worried about those. Those typically aren't even reported. They could say it's a variant of uncertain significance, meaning it's in the middle. We're not exactly sure, is it benign or does it actually cause that gene to not function? The lab can't say with certainty. So they say, we don't know. We're going to leave it up to you guys to figure out. And then on the other side, it can be likely pathogenic. Pathogenic meaning that gene's not working. This is what is called, this genetic change is causing this gene to not function, or definitely pathogenic. So there are some uncertainties when we interpret genetic testing results. Usually to make a diagnosis, the variant is usually pathogenic or likely pathogenic, but there are probably some cases out there where people get a VUS, and then it's kind of up to the clinical team to do more research and decide, do we think this is the cause of these symptoms or not? Again, it's normal to have genetic changes. We all have genetic changes in our bodies. That's why we're all a little bit different. Some are inherited from our parents and some are brand new in us. And again, it's why we're all have those mild differences. It's normal. We expect variation. We are more alike than not, but we all do have small variants, and it's normal to have that. Just a matter of, did that person develop a variant in a gene or in a location that caused that gene to not function how we need it? And that's kind of how we look at HADDS. For some reason, there was a change in the EBF3 gene, and that gene's not functioning how we need it to now. I also kind of looked at the types of variants. I get this question quite a bit from my families who have different types of genetic conditions and who are typically involved in a really great family community like you guys. They start talking about types of genetic variants. So I wanted to touch on that briefly. There's different types of genetic changes that you can have that can cause that gene to not work. So if you think about, if there's a sentence, we typically read genes in sets of 3 letters. So you think about a sentence that says the cat had red fur and ran far, period. That would be the gene. But you can have a missense change where one of those letters gets changed. So now it says the mat had red fur and ran far. That doesn't entirely make sense. I would actually consider this to be something like a variant of uncertain significance. It's hard to tell, is this single letter change actually changing the meaning of that sentence? The mat could have red fur. I don't think the mat could run far though. So how is this interrupting the gene function? So these are missense variants are some of the hardest to understand. They can be nonsense, meaning you end the sentence too early. So just be the cat had, period. A lot of times nonsense variants typically do cause that gene to not function as we would expect. You can also have things called a frame shift, where you essentially remove a letter or add a letter, and it throws off those 3-letter codes. And so that gene, that sentence doesn't make sense anymore. Similar, you can also just add in things. So now we added in the word fur twice. If you type on a Microsoft Word, they're going to red squiggly line that because it doesn't want it to be there. So again, these are kind of what we think about when we're thinking about types of genetic changes. Again, we don't have any control over what type of genetic change it is, why you got it, how you got it. But sometimes we lose these things to help us understand. And it is a kind of a common question I get from families who are starting to understand a little bit more. There's a couple of other types of changes that have been reported. So things like a whole gene deletion, or just for whatever reason, that entire EBF3 gene is missing. And in those cases, it's sometimes you see that The genes flanking it or on other side of that EBF3 gene can also be missing. So those presentations sometimes look a little bit different. Or you can have things called splicing variants, which change the order of how a gene is or a protein is assembled. And those can also, you can see those too. So how did we get to all of these, right? I would say most people in here have some familiarity with genetic testing. So you can do things like looking at just one gene. So you can look at just the EBF3 gene, which is typically what we do for a family after we have a diagnosis. If you want to test other family members, we just got to look at that one gene. Or you can do more extensive genetic testing, things like a panel. So maybe you look at 20 genes at the same time, or in a lot more cases, you look at like 2,000 genes at the same time. You can have big gene panels that are maybe for a patient who has hypotonia, for example, which is a feature I know of HADDS. So you would test for all the genes we know that can cause conditions with hypotonia, for example. I would say a lot of you maybe were diagnosed with exome sequencing though. And so that's just reading all of the genes. That's reading the entire encyclopedia set, looking for any spelling changes in any of the genes that could cause one of those genes to not function. The next step beyond that, which you might start hearing about, is genome sequencing. It's something we're starting to do, but genome sequencing is like reading that encyclopedia, but then also reading all of the footnotes. It's also reading the dedication page in the front that tells you where it was published by what publisher and what year. And so it's sometimes hard to interpret because I don't know what to necessarily do with that front cover page information, but it's reading all of the extra material that aren't genes. Yes. So yeah, chromosome microarray is not included on this because we're talking about sequencing, but you beat me to it. The other technology we talked about is the chromosome microarray, or CMA, and that is specifically looking for missing or extra genes or pieces of genes. So when you have something like the entire EBF3 gene missing, that's a lot of times diagnosed by CMA because it can look, that's all the CMA does. Its job is to look for extra or missing chunks or pieces or words in that encyclopedia. Great question. Okay. So I have done a really good time on my time. So kind of just going back through what we talked about, I want to kind of make sure we hit on like my take-home messages. So HADDS is caused by changes in the EBF3 gene when that gene's not working as expected due to a genetic variant. We talked about different types of genetic variants and different ways to come to that diagnosis. The recurrence risk for something like HADDS can range from about 1% all the way up to 50%, depending on if that variant is present inside a parent or not. And that risk or that number can be refined by further genetic testing or even doing things like testing before pregnancy, prior to conception, or during a pregnancy. And we can talk more about some of those reproductive technologies if there's interest. And then otherwise, I think the other thing that I didn't really say, that is, I think most people in this room have probably been told, again, we all have genetic variants. If your child has a brand new genetic variant, it's nothing you did, it's nothing you didn't do. We couldn't have changed it. We couldn't have caused it. We couldn't have predicted it. It's never anyone's fault. I spend a lot of time talking to parents who sometimes question themselves about why this happened. And that's a really good question. It's one that keeps you up at night, but it's not the influence of an external factor or environmental factor that someone experienced during pregnancy or even after pregnancy. It's something that was inherent to that person and it makes them special and unique, just like all of us have random genetic changes as well that make us unique as well. Okay, I left a lot of time for questions because again, this is kind of a broad topic and there can be very specific questions. I'm happy to take them from the chat, but if you have a personal question too about your child and want some more specific, oh my gosh, want certain information about your genetic result or anything, you can always shoot me an email or you can ask someone throughout the weekend. I'm also going to stay after for a little bit too, so I'm always happy to talk to you guys one-on-one as well. Okay, and I'm happy to take some questions. I see a question in the back.
You were just saying that if neither of the parents has the genetic variants, then there's nothing that you did or didn't do or anything. By any chance, is there a possibility that it skipped the generation?
Yeah, so the answer is no. We would expect to be able to detect it in a person, and someone who has that genetic change, we expect them to show symptoms. So it's something we should be able to clinically see, and things like dominant conditions, at least in HADDS, we don't see it skipping generations.
Thank you.
Yes.
What we said was a de novo.
Mhm.
It was only him that we had nothing. It was only for him. Is it possible? Because I think
Yeah.
it's
So
inherited.
So the question for anyone online who didn't hear at the beginning, so she said that you and your husband had genetic testing along with your child, and it was
Yes.
determined to be de novo or brand new in your child. That is the case for a vast majority, and I would say most people in that room are going to be in the same boat. That is how we see most of these genetic conditions, and even outside of HADDS, a majority of the conditions that I diagnose for various other genetic syndromes are de novo in a child and brand new in that child.
Should we both be tested like deeper, like with something bigger, or no?
There's no indication to do deeper testing. If you have had a genetic test and it was normal, or you didn't carry that variant, no further testing is recommended for a parent. It's most likely de novo, but again, we could do all the testing in the world and we could still figure out you don't carry it. So no further testing are typically recommended for parents. If you're planning a future pregnancy, I would encourage you to talk to a genetic counselor just about any of those risks, or about if you want to do any additional testing during your pregnancy, but no more tests are recommended. Dr. Wengler.
No, yeah, I would just add the 1% number that you gave would be the upper limit. That'd be like the
very
worst
top.
case scenario that we would give a family like yours.
I usually
And
say
it's
less
based
than
on the idea
1%.
of germline mosaicism,
Mhm.
which is I can't detect it when I test you, but perhaps more than one sperm, more than one egg has the change. And the 1% comes from years of experience with many genetic disorders with these same mechanisms. And there's no reason to like biopsy, other tissue, because it's very little would change our estimate of that 1%. Then the 50% is like if we had detected mosaicism in one of the parents, then the 50% would be the worst case scenario for that family. So it's a little confusing, but that's the idea.
Yeah, there's two behind you. Or do you want to do it on?
We have
chat?
a
Okay.
virtual
Great.
question. Give folks time to run microphones around. This is virtual from Deepa Babington. Can you talk a bit about how much having a genetic change on something like the EBF3 gene determines outcomes in life? For example, can two people with the exact same genetic change have different health consequences? Is it possible there are people walking around with EBF3 change who have no health consequences?
Yeah, very good question. So the first part of that question, the answer is yes. Two people can have the exact same genetic change and have a different clinical course or present with slightly different symptoms. That is seen very commonly across really any genetic condition. And that's because the rest of our genes are unique to us too. So yes, we do see that very commonly. And that's why when you get a genetic diagnosis, you can't say with any certainty your child or that person's definitely going to have A, B, and C. We can say they might have symptom A, might have symptom B, they might have symptom C, but we can't tell you exactly when something will present or if that will be present. I think the second half of that is it possible there's someone walking around with a change in the EBF3 gene that just doesn't know because they don't have any clinical symptoms? It's unlikely. I'm not going to say impossible, but it's very unlikely because, again, we expect someone who has a change making that gene not work to exhibit some degree of symptoms if they truly have that condition. And I think we had some questions back here.
Sorry, you just answered part of my question. It was about can people have it that don't present symptoms and stuff? Because I'm wondering what happens after my son, say, he has a child seemingly normal, no gene variation. What about that child? What about his grandchild? what we doing next generation?
Yeah, so let's say so your child who has HADS has a child, that child was tested, they don't have it. That child's risk to have a future child with HADS is the same as the general population risk. It goes back to the same as any other person who doesn't have a family history of HADS. Yes.
My mum was undiagnosed for 50 years, so it's possible for someone to walk around knowing they don't have it.
Yeah, I mean, I think that's also something we see in a lot of clinical genetics. I've diagnosed several parents who have more mild symptoms and just weren't diagnosed because genetic testing and things like this, I mean, we didn't even know about HADS until very recently. So people can have undiagnosed conditions, and especially if they had mild symptoms, and not know until later.
Thank you.
Is there across the board, I guess, ethics or things surrounding testing siblings that we don't necessarily have a specific reason to test them, like if the parents aren't tested and that kind of thing? Because that was something we ran into, because there's concerns about, the child finding out they have it and that kind of thing. What is I guess, you guys or the genetics community say about that?
Yeah, in general, the genetics community says you shouldn't test a minor for something like an adult onset condition or something that won't affect their health until they're an adult. But in something like a genetic condition like this, or another type of condition that does have a pediatric onset and pediatric symptoms, as long as everyone consents to testing, it can be appropriate to test siblings. Even if that sibling's healthy, like we've been talking about, it's hard to predict symptoms. Or, if a family has a child and they're very young, they just haven't displayed symptoms, it's acceptable or it's okay to test that seemingly asymptomatic child, given that there is a slightly higher risk that they could be affected. Yes.
Another virtual question from Heather. I was wondering, since my daughter has the genetic change, she has a deletion. My husband and I did not carry it. I was wondering about the chances of her having a child one day with the same deletion if her partner did not have a genetic mutation.
Yeah, so it's still going to go back to that 50% risk. So deletions or single gene changes in this case are the same. So again, her child could pass on the chromosome 10 with the deletion, the chromosome 10 without the deletion, and it's a 50-50 chance in any pregnancy. You asked insightful questions. I appreciate it.
I've got one more virtual.
Okay,
Is there someone? We go.
Oh. For this, did you say the majority are the ones that they're not just passing on from their parents? It's really just popping up. That's
yes,
yeah.
vast majority, almost all. It is something brand new in the child, or de novo is the word that we use in genetics. De novo in the child, or it's not common at all. It's pretty rare to have a parent who is mosaic or themselves mildly affected and not previously diagnosed. It's almost always going to be brand new. You had an online question.
Okay, this is a virtual from Camilla. Our diagnosis came after exome before we did microarray, but the results were normal. Why did it happen?
The diagnosis, so was the follow-up question, was the diagnosis made via exome or CMA? Can you read it one more time?
Our diagnosis came after exome.
Okay.
Before we did microarray.
Okay.
But the results were normal. Why did it happen?
So if the CMA results were normal, that is because the CMA only looks for deletions and duplications of genes. So it sounds like this person didn't have a deletion of the gene. And so an exome then was the test that was used to actually read through that gene to find that spelling change. So CMAs and exomes look for different types of changes. So it sounds like that child had a change that could be detected by exome, but not by CMA.
How might people who want to meet with a certified genetics counselor identify somebody?
Great question. We would love to see all of you. So there's the National Society of Genetic Counselors, or NSGC. We have a Find a Genetic Counselor tool. So you can search based on where you are located. And like you can also search by specialty. So you can search by like general genetics, adults or pediatrics, or even prenatal if you want to meet with one. And it will pull up all the genetic counselors in your area. You can meet in person locally, but there's a lot of genetic counselors who are certified to do like telehealth consultations across anywhere in the country. If you are international, it's a little bit trickier. Several countries do have their own Society of Genetic Counselors, and so you can try to get in touch with that. Or you can try to get in touch with someone from the US or the National Society of Genetic Counselors, so we can see if we can, anyone has international connections. That's where I would start.
How rare is the gene of the world?
Do we know what the incidence is? Do we have a guess? Up to 5,000 new diagnoses a year.
For EBF3.
Yeah, so we're not entirely sure how common this condition is, and that's because it's pretty new.
Yeah.
But it's something we're trying to figure out, and we're making diagnoses of it more frequently now that we know more about it. Good question. We don't quite have the answer, so you're gonna have to keep coming and asking that question.
Thank you.
Okay, one more. Do we know how many have been diagnosed to date? It's hard. That's a hard thing to know. Do you know how many families are in this group? Okay, we're going to estimate more than 300. It's one of those things where you have to, like, lots of people get diagnoses, but unless we report that diagnosis to someone or they join a group like this, there's not a good way to track them. But more than 300 is good. The last one of the papers I read was only 50 people, so making good progress. Yeah. Okay, last
virtual question. Again from Deepa Babington. Can you talk a bit about deletions? Is it correct that a partial deletion, for example, generally produces milder symptoms?
I think we're still working out genotype phenotype correlation. So we don't have a straightforward answer to that. Just like we were talking about different types of variants, we haven't been able to tease out if a certain type of variant, including something like a partial gene deletion, results in different severity or different certain symptoms. That's something we're trying to figure out, but it's a work in progress. You usually need a lot of people to better understand that with lots of different genetic changes. So not a good answer yet, but it's something that join us for research things and we're going to try to figure out. Okay, thank you.
Thank you so much.
Of course.